Thank you for this invitation to make a presentation on the COVID vaccine and vaccinations. Uh, Many thanks to the Faith and Science group in biology who have extended this invitation to me to be able to do the presentation. Let me just share my screen and see what we'll be talking about. Um, Several things that I want to just outline here, some questions that I want to talk about. First, I want to ask the question, what is immunity, vaccination, or immunization? And then look at how vaccines are made and how they protect us. I'll also talk about the central dogma of biology in relationship to some of the vaccines that are being made, and then look at the role of the SARS-CoV-2, the cause of COVID-19, the genome in the vaccine production, and then we'll point out some of the vaccines that are currently being administered for the COVID-19 disease. By way of introduction, I just want to look at first at just how the body defends itself against infections, especially against pathogens like viruses or bacteria or parasites and so on. We can divide the body's immune system into three major categories we normally refer to as the first line of defense, and then the second line of defense, and lastly, the last line of defense. The first line of defense involves the innate immunity. Um, the innate immunity is just the immune system that we are born with right from the beginning. So things like the skin, every baby has a skin. Things like acidic environments. For example, the stomach is highly acidic. Some of the secretions that we secrete, whether it's on the skin, the mucous membrane, some of the secretions again, these are all first lines of defense. They protect us against pathogens from the outside. Now, if a pathogen manages to breach these external barriers, then it encounters some internal defenses, which are still part of the innate immune system. Things like phagocytic cells, these are cells that just hunt pathogens, engulf them, and destroy them. Defense, natural killer cells, some proteins that are produced to destroy pathogens, and then also the inflammatory response. All these are part of what we call the first line of defense. This line is very rapid, it's very fast, protects against a broad range of pathogens, but there is no memory. So the body will respond the same despite the different types of pathogens that it encounters. Now, when these first and second line of defense mechanisms are breached, if the pathogen then manages to get into the body and into circulation or into the cells, then the pathogen encounters what we call the adaptive immune response, or the specific immune response as it's sometimes referred to. And it falls basically into two major categories. The one which defends against pathogens that are in bodily fluids like the blood, lymphatic system, and so on. And then those pathogens that enter the cells, these are inside the cell. And so the body has to find out which cells have been infected and be able to deal with those pathogens. And of course, the defense against pathogens in the fluid normally involves the production of antibodies, two major types, the IgG and IgM. And also the defense against pathogens inside the cell will involve cytotoxic T cells. These are cells that recognize infected cells or affected cells and destroy them so that they don't serve as small factories for the multiplication of pathogens. Now, the third line of defense tends to be slower. Most of the times, the response takes a few days to a few weeks in order to become fully active, and it normally recognizes specific pathogens. So antibodies produced against COVID-19 virus, for example, are not going to recognize the influenza virus because these are specific responses. It's important also to note that this specific defense mechanism also keeps a memory. So after the pathogen has been cleared from the body then keeps some cells, memory cells, that remembers the type of pathogen that initially stimulated the production of whether it's the antibodies or the cytotoxic T cells. And this memory is what is important when we're looking at vaccinations and vaccine production. Looking at vaccines normally just tend to be small pieces of the pathogen which stimulates antibody production. So if you recall, we said the third line of defense is the production of antibodies. So when a pathogen enters the body, it stimulates the immune system. The immune system starts to produce antibodies. These antibodies can then interact with the pathogen in a process that can result in neutralization, which is what is indicated here. So you have a virus, for example, which is coated by antibodies. And because it's coated by these antibodies, it cannot interact with the cells, and therefore it fails to invade the cell and fails to cause disease. The same thing with the bacteria. So this process is important because it helps the body to get rid of pathogens. Now, when we look at the way the immune response, the immunity works, remember I said that when the body encounters a pathogen, it can produce antibodies. And now I want to look at this diagram here because it has a bearing on what vaccinations are all about. The first time we encounter a pathogen, say for example, the COVID virus, the body takes a bit of time to try to figure out what type of pathogen it's dealing with, and then it will start producing antibodies. And these antibodies will normally peak after about 14 days from the first initial contact. So if you're looking at the number of antibodies that are being produced in circulation, that these antibodies will peak about 14 to about 20 days after production. And of course, during this time, they're encountering and interacting with a pathogen, and in the process, they are neutralizing the pathogen. And eventually, the pathogen is cleared. But notice what happens here. We started off with almost no specific antibodies. At the end, we're remaining with some level of antibodies in circulation. And this is what leads to the memory that I mentioned. The body is remembering the pathogen that initially stimulated the production of these antibodies. So that if the body then encounters the same pathogen a second time, it's not going to take so many days to respond or to produce antibodies in large amounts. The response is normally much quicker, and it's also more aggressive because the number of antibodies that will be produced in circulation is much larger, more aggressive, and so neutralization tends to take place very quickly. And this is the protection that arises from the second encounter with the pathogen. And so when we're looking at vaccinations, what we're basically doing is to introduce a pathogen to the body so that it can encounter that pathogen and start producing these antibodies in the first instance, so that the next time the body encounters the same pathogen, it can respond more aggressively and be able to deal with a problem much more quickly and in a more aggressive fashion. So vaccinations, basically what we're doing is we're introducing pieces of the pathogen and introducing the body to the pathogen so that it can start the process of producing the initial antibodies and keep a memory of those antibodies in circulation. And immunization is just the result that is obtained after the immune system has been stimulated. So this is the whole basis of vaccinations. You want to start the process so that the next time the body encounters the pathogen, it can respond more aggressively. When you look at the central dogma of biology and also viral infection, normally in the body, the cells in the body have a nucleus, and then out of that nucleus, you have the cytoplasm. The DNA in the nucleus is normally transcribed into messenger RNA, which is then transported into the cytoplasm, where the ribosomes, which are just small machines that make proteins, will make the proteins. So the flow of information normally is in one direction. And notice that some of the pathogens will also have similar structures like RNA. So for example, COVID virus has an RNA, which if it entered the body, these small machines, the ribosomes, can still recognize that RNA and use it to make proteins. Except the proteins that are being made, these are viral proteins. These are then assembled into viral particles, which can then exit the cell. And this diagram is just illustrating that. And so the central dogma that we're talking about, which is what normally happens in the cell, introduction of foreign RNAs can also lead to the same process where the RNA is recognized and used to make proteins. And so when we talk, for example, in the making of vaccines, and we talk about RNA vaccines, what we are doing basically is to introduce RNA into the body and let the body's machinery utilize that RNA to make proteins. And some of these proteins will be viral proteins. And because they are foreign proteins, the body's immune system then will respond and produce antibodies in order to start the process of clearing those antigens or pathogens. Looking carefully at the genome of the coronavirus, the SARS-CoV-2, the genome is actually a very simple genome. The genetic information, it has some genes that code for non-structural proteins. These are proteins that are used by the virus to replicate within the cell. And then it has other genes that code for structural and accessory proteins. So for example, like the S protein, which are the spike proteins, these proteins that you find on the surface of the virus, these are normally used to identify receptors in the host. So this would be a human host, like the rest in the lungs. You have the SCE2 receptors, which are recognized by the spikes. The virus then binds to that and allows it to start getting into the human cell. There are other proteins as well that you find, for example, envelope proteins, the membrane proteins, and so on. These are proteins that can be used to recognize the pathogen. And so some of the vaccines that are being produced will be vaccines that introduce the spike proteins in the absence of the virus itself. Okay, or alternatively, you introduce genes that can be used to make the spike proteins so that the body can make these spike proteins and then stimulate the immune system. And moving on to the SARS vaccines, SARS vaccine development has fallen into two categories. There are what we call the classical vaccines. So for example, some viruses, viral-based vaccines where you take the virus, whether it's the COVID itself, kill it, by inactivating it, maybe treating it with chemicals or UV light and so on, so that it's now dead, it cannot reproduce, and then introduce the dead virus into the body. Now, this dead virus still has these antigens on the surface, so the body can produce antibodies against these antigens, and hence will start the primary response, so that the next time the actual virus infects the body has already encountered some of these proteins on the surface from the dead virus. In other cases, you can use the live attenuated viruses. These tend to be, they are normally the closest thing to the real thing, where you take the virus itself, genetically engineer it so that it cannot cause disease, but it still has features of the real thing on the surface. And then you introduce that into the body, and then the body then starts to produce antibodies against some of the structures on the protein. You can also just purify the proteins, the surface proteins, the spikes, and introduce these as vaccines into the body. This is another way that can be used to produce vaccines. This is what is referred to as the subunit vaccines. Alternatively, you can use viral particles, where it's basically an empty virus with surface proteins. You introduce this structure into the body, and the body then is stimulated to produce antibodies. Now, these processes tend to be very slow, and in many cases they require the growing of viruses themselves, and so you need stringent growth conditions. You also need specialized lab like the BSL 3 labs. This is biosafety level 3 labs, and they also need to undergo stringent testing. So in many cases, these production of vaccines using these classical methods tend to be very slow. However, there are other, what we would call the next generation vaccine development efforts. In this case, you're using platforms that don't need the virus. So in this case, you just need the genome of the virus. And you can easily adapt different platforms that have been used for different vaccine productions. For example, production of the influenza virus. You can use some of the information that you've gained from production of these other vaccines and apply them very quickly to COVID vaccine production. To give an example, the COVID genome became available in January 2020, and the first vaccine trials were started in March 2020. Just a few months after that, people had already started making vaccines. And these next generation vaccines are based on several things. The first one is the use of viral vectors, where you take the genome of the COVID virus, take some of the components like the spike proteins, introduce them into a virus that we already know, which, for example, the adenovirus, which you've removed most of the genes for the virus, and use the viral particle as a delivery vehicle to deliver the genome of the spike protein from the COVID virus into the body. And then as this viral particle enters into the body, it introduces the genome for the spike proteins, and the body starts to make these proteins, and hence to stimulate the immune system based on the spike proteins that have been used. These tend to be very efficient, except in many cases, the viral particle itself can also have its own, it can also stimulate production of antibodies based on the virus itself. And so you have to now start distinguishing between the viral particle antibodies and the vaccine antibodies as well. And there are two types, the replicating and non-replicating viral particles, and I don't need to go into this. The other type would be just the DNA, then a naked DNA introduced in a plasmid, and you can introduce this into the body, and this plasmid also carries genes for which code for the spike proteins, and once they get into the body, then the body uses this DNA to make RNA, and then RNA is used to make proteins, and these proteins will be spike proteins. The other type is where you basically just taking the RNA instead of even using the DNA. You just take the RNA and package it into some particle and introduce it into the body. In this case, the RNA is straight away recognized by the ribosomes. Those small machines that I said can be used in the body to make proteins, and the RNA is then converted to proteins, and then the proteins will stimulate the immune system. Now, this is again falls into two major categories: just the simple messenger RNA and the self-amplifying RNAs. Again, we don't need to look at that. The only problem with the RNA viruses is that RNA is very unstable, and so you normally have to introduce delivery vehicles that will kind of stabilize the structure, and many of these delivery vehicles require very low temperatures to keep the RNA stable. And so these delivery vehicles will then introduce the RNA into the cell, and then once inside the cell, the delivery vehicle is removed, and the ribosomes can be used to make proteins, and these proteins then can be utilized as antigens to make proteins. Okay, now there are several different vaccines that have been are being developed. Some of them are based on the next generation, the genetic type. There are those that are based on the protein components like spike proteins, viral particles, and others. And these are just different types of vaccines that are in development, or some of them have even been approved yet. And this is the same thing. Some of them are based on viral particles, others on just the DNA, and some of them on the messenger RNA. And coming to some of the vaccines that are currently being used and those that are promising, some of them fall into the killed viruses. I think I talked about that. So there are several of them that have been approved, for example, for use in China, India, and so on. And then there are those that are based on the purified virus components like spike proteins and so on. Again, there are some which are in phase 3 trials. And lastly, there are the non-replicative viral vectors, where you take the viral vector and incorporate the spike protein. And so the AstraZeneca virus vaccine, for example, is based on some viral vector, and this has been approved for use in Canada and some other countries. And then, of course, there are those that are just simple messenger RNAs. So the Pfizer vaccine, the Moderna vaccine, these are just simple messenger RNAs that are packaged into some viral delivery delivery vehicle and introduced into the body in order to stimulate the production of antibodies. Of course, the use of adjuvants, I won't go into that. Normally, the pathogen itself or replicating viral vectors don't need the introduction of adjuvants. These are just components or antigen molecules that are added to the antigens in order to allow the body to stimulate the immune system. And there are many different types of adjuvants that are being used, and we don't need to go into that. Okay, just some papers that have been used in the preparation of this presentation. This is a good paper which gives a very good summary of some of the advances that have been made in the different platforms that are being used to address the production of vaccines. This is another one on antivirals and gene therapy against emerging coronaviruses, the use of adjuvants, and so on. Thank you very much.
Thank you, Anthony, for laying that groundwork. Just let me click a button or two to get this sorted so I can see myself at least as I talk. So my task here is fairly straightforward. Anthony did a good job of laying the groundwork of how the body fights infection and how vaccines allow the immune system to see a pathogen or parts of a pathogen before actually encountering an infection. My task is to describe how vaccines go through the process of development and approval. So Anthony got the exciting biology and I get the boring bureaucracy. But it's important because one of the overriding concerns that many people have, and many Christians as well, is questions along the lines of how safe are these vaccines? How well can we trust the process that produced them? And also, this happened very, very quickly. Does that mean that corners were cut? Those kinds of things. And it is true that normally vaccine development is a slow process, and it's spaced out over a long period of time. And these different steps that we'll go through to describe how a vaccine comes into being and is approved will illustrate the normal process of how long it normally takes. What was done in the case of COVID-19 vaccines is, due to the urgency of the pandemic, all of the normally in between each of these steps that I'm about to describe for you, there would be long delays, and they would be strictly regulatory delays, or just because of lack of urgency, perhaps funding delays or things like that. The urgency of the COVID pandemic has basically taken away all of the superfluous time. We've basically compressed vaccine development into the shortest time allowable, while maintaining the rigor and the scientific standards that protect us and to make sure that vaccines are safe and have high efficacy. So that is the goal. The goal is to design of vaccine development is to minimize the risk involved and also to maximize the efficacy. We want vaccines that are safe for people to take, and we also want vaccines that are effective at fighting and preventing fighting the pathogen and preventing disease. So let's just walk through some of the steps that are required for vaccine development. As an aside, a really nice outline that, I mean, this information is available in many places on the internet. One nice spot here is this history of vaccines. It lays it out in nice layman's terms, where you can see. Now, the process is going to vary a little bit from country to country. This site here is very US focused. The US is a major world leader in vaccine development through the National Institutes of Health. But the European way of doing things is very, very similar. So I've generalized the information when I say like regulatory bodies. That might be a different body in Europe. It is a different body in Europe relative to, say, the FDA in the United States. The way this normally starts, vaccine development normally starts, is through what are called preclinical studies. Preclinical studies are when scientists get interested in a pathogen, or they learn about a new pathogen. So that leads to just basic scientific research that looks at this pathogen that hasn't perhaps been studied well before, or it's a new pathogen. And the study of that pathogen usually is to identify what sort of pathogen it is. In this case, we're talking about viruses, and it's a new virus with COVID-19 that we're interested in. And studying that virus and looking at what antigens it might have that would be useful to target. So antigens are just the components of the pathogen that would be useful to have an immune response against. Anthony did a good job of describing that. What's really interesting is, in the case of COVID-19, and generally when we do vaccine development, we benefit hugely from the fact that there has already been a large amount of basic research done on very similar pathogens. So we were already well on our way to understanding COVID-19 because we had done, and when I say we, I'm using science sort of as a collective we here, the scientific community had already done a lot of work studying various coronaviruses. And that work allowed us to, understand their biology, understand how they replicate, understand that they have an RNA genome, that sort of thing, understand that their spike proteins on the surface is the key thing that they use for attachment to human cells, and that's a key target to try to raise antibodies to, all of those sorts of things. Another thing that was really important for preclinical studies, actually, just as an aside, all of that, of course, precedes us discovering COVID-19. We've studied coronaviruses for many, many years. Technologically, there's also advances that have been made in vaccine development that were very useful. So Anthony did a good job of talking about RNA vaccines and how they can be administered directly as RNA. The lipid nanoparticles that you put the mRNA into to allow it to actually enter cells and be an effective vaccine, that technology was not worked out from scratch for a COVID-19 vaccine. That technology is also something that was available and had also been already tested for safety. So that was a useful tool to sort of take off the shelf, as it were. So this led to a very rapid set of preclinical studies. We already knew a lot about coronaviruses. We had certain technologies that had been tested for safety with the lipid nanoparticles. And that is what allowed very rapid preclinical trials to be done for the mRNA vaccines. Now, the same sort of things apply for the AstraZeneca vaccine using an adenovirus vector, like Dr. Siame mentioned. We have done many vaccines using adenovirus vectors. So that technology was sort of ready to hand. Typically, a preclinical study would be something that would develop a proposed vaccine and then would be tested in some way, perhaps on cell lines, so on cells that are in tissue culture, or in animal models to see how well those animals, such as mice, for example, would have an immune response to that. So that is typically done by scientists, different research groups. They would publish their results in papers. Those papers would be reviewed and vetted by other experts to make sure that those experiments had been done correctly. And then if that all goes well and there's a perceived need, then the next thing that would happen would be an application for a phase 1 trial. So a phase 1 trial, as you'll see, is when a small number of healthy people are given the vaccine. And the main point of that is it's a very small number of people, like say 20 to 50 or so. They receive the vaccine, and the primary purpose of them receiving the vaccine is not necessarily to test for its efficacy, although they will be tested for an immune response. But phase 1 trials, one of the main focuses of a phase 1 trial is to make sure that there are not adverse effects. Now, it's a very small sample size, so it will have relatively low power to assess that, but it would flag up if there are any very common adverse side effects. The point to make is that a lot of people think, okay, you know, phase 1 trials are how these things get started, but there's already been a lot of work prior to a phase 1 trial, and there's also been a lot of regulatory eyes on the research prior to a phase 1 trial through the application. So the regulatory boards would review the preclinical data, and they would decide if that data has been collected appropriately, it has been controlled appropriately, and then they would decide if they would allow for a phase 1 trial. Once you have a phase 1 trial, and if it is successful, we don't see adverse effects, healthy people don't get sick when we give them something. As an aside, these different phases of trials also apply to drugs, not just to vaccines. If a phase 1 trial goes well, the next stage is to apply again for a phase 2 trial. And it's the same process that we just went through. So a regulatory board again would look at the data from the phase 1 trials, and they again would decide if it had been done correctly, and they would decide if a phase 2 trial is warranted. They would be primarily concerned with the safety of the drug or vaccine at that point, that it's not generating an adverse effect. If a phase 2 trial is permitted, then when that goes ahead, we now would have a larger number of healthy people who, again, it's very similar to a phase 1 trial, but basically what we're doing is we're just upping the sample size of the phase 2 trial. Again, the focus would be on safety and looking to see if there are any adverse effects. With the larger sample size, that gives greater power to detect events that are less common, just based on probabilities. And of course, in the case of a vaccine, phase 2 trials are also used to collect data to see how much of an immune response is generated. And that is basically after the immunization protocol and time to develop those antibodies, then there would be testing to see how good the antibodies are that are being made, how well they stick on to the antigen that was put in the first place, and then just how much of them are being made, like what the concentration of those antibodies are in the people who have been vaccinated. If that all goes well and adverse effects are not seen, then there would be an application for a phase 3 trial. Phase 3 trials tend to be the trials that people hear about because these are the large trials that involve tens of thousands of healthy individuals. Now, but again, there's an application for a phase 3 trial. Regulatory boards again review the data from the phase 2 data, and then they decide if a phase 3 trial is warranted. A phase 3 trial, the primary interest in a phase 3 trial, of course, is to address efficacy. How well does this vaccine protect against COVID-19 in this case? And this is where you see those numbers for different vaccines, you know, Pfizer at 95, Moderna at 90, whatever it was, similar to the Pfizer. The AstraZeneca one, lower numbers. So this is simply done by having tens of thousands of individuals who are in a group that receive the vaccine and tens of thousands of individuals who are in the control group. They do not receive the vaccine, but they do receive a placebo. They go through the same process. They get an injection. And an important thing about all of these trials at this point, aside from phase 1 trials and phase 2 trials, actually, there's some variability about whether or not they're blind or not. But phase 3 trials are what are called double blind. None of the participants know if they have an active vaccine or a placebo, and none of the individuals administering the shot know if they have active vaccine or placebo. And this is the gold standard of trying to figure out if there is statistical difference between the group that receives the vaccine and the group that doesn't receive the vaccine. So in order to make that judgment call, there needs to be a number of infections. People actually have to get COVID in that large group. And there needs to be a comparison between the control group and the placebo group to see if there's a statistical difference in between the two groups that can be attributed to receiving the COVID vaccine or not. If phase 3 trials are successful and adverse effects are not noted, or if they are noted but are below an acceptable threshold, and then there would usually be protocols put in place to be aware of those and to try to minimize their negative effects. Then regulatory boards, again, there would be different in Canada or in the States, but you would have noticed that many of the vaccines as they went through this stage were approved for use in the United States and then in Europe and then Canada within a short order because the processes are very similar in the different countries. Regulatory boards, again, made up of experts, review the phase 3 data, and they decide if the data has been collected appropriately and controlled appropriately, and if the vaccines are have the efficacy that we're looking for. Now, after a phase 3 trial, that's not the end of the story. And actually, we've seen this with AstraZeneca, as you've noted, most likely. After approval and release, as people are immunized, now this is not double blind, of course, at this point. At this point, people know that they are getting an active vaccine, and the people injecting them know. So placebo effects are now no longer under consideration. But as more and more people get a given vaccine, that is a further opportunity to study its efficacy and also to study adverse side effects. So this is what is called post-approval monitoring, and it's sometimes called phase 4 trials. Phase 4 trials would be a more formal study done by the people who have made the vaccine to continue to monitor its efficacy or adverse effects. More generally, post-approval monitoring continues, aside from that formal phase 4 process, but they're very similar. We're just, as the sample size increases of different people getting these vaccines, we continue to watch and we continue to track whether or not there are adverse reactions taking place, and as well as efficacy. And the blood clotting issue with the AstraZeneca vaccine that we've heard about in BC because it was news here, that is an example of post-approval monitoring. So even though AstraZeneca is approved for use, as more and more individuals were given that vaccine, it provided greater power statistically to detect more rare events. And there is a possibility at this point that the blood clotting issue may have statistical significance. That's still something that's going to be studied, and greater sample sizes are going to be used. But because of that possibility, you'll notice how careful the regulatory bodies were, that there was a pause on delivering that vaccine to certain age groups because of the possibility that there was a statistical significance for that adverse effect that had not cropped up in the smaller phase 3 trials, although smaller, I guess, tens of thousands of people. But that shows you how rare this particular event was. Okay, that's my slides for that. And then later on, I will talk about Christian responses and discussion points there, but I will hand it off from there.
Thank you, Dr. Venema. I will continue on here. Thanks to Sam and to the Faith and Science group. I am going to do this old school, no fancy PowerPoints, and actually I'm going to read my presentation. So let me get right into it. This is the Faith and Science Club, so let me begin my talk with the faith piece before moving into the social science piece. I'd like to read from Matthew 11, verse 27. No one knows the Son except the Father, and no one knows the Father except the Son, and those to whom the Son chooses to reveal him. God chose to dwell among his creation in the person of Jesus Christ. Christ, as fully God, forever in relationship with the Father and Spirit, came to reveal the Father to us. Perichoresis is a Greek term used to describe the intimate nature of the relationship between the members of the Trinity, the oneness and the threeness of God. Christ, through his death and resurrection, which we have just celebrated, is inviting us into this same oneness. In this Trinitarian relationship, we see how the parts and the whole of a relationship function in perfect harmony and have for all of eternity. In that relationship, I find both my basic interest in sociology and hopefully a transition to our discussion tonight. Sociology is a discipline that seeks to understand how human behavior by looking at the intersection of the individual and the group, individual human agency and larger social forces at work found in the institutions of politics, education, religion, economics, and the medical fields, for example. A vaccine as a tool of public health is an excellent example of the dilemma of individual rights and social needs. The challenge of the coexistence of the one and the many are being lived out today. Let me ask you a question. Is the COVID-19 vaccine being promoted at the individual or the group level? Does 95% efficacy against serious illness, hospitalization, or death appeal to the individual more than to the group? And what if the hoped for reduction in transmissibility doesn't fully materialize? What if the vaccine reduces serious effects in those vaccinated but does not act as a prophylactic to the transmission of the virus? Recent data seems to support this hoped for reduced transmissibility, but that's not what the vaccine's main selling point was. Something to consider as we reflect on the personal versus the social benefits of the vaccine. I believe we'll see both, but we're at a point in time where the studies continue. In the time remaining, I want to provide a brief overview of 3 sociological theories or lenses that are used to try and better understand, predict, or explain human behavior. I plan to provide a brief example or 2 germane illustrations to vaccines, but maybe not limited to them. It's not my goal to promote or discourage the use of vaccines. Some may find that unsettling on both sides of the issue. But it is my goal to provide a high-level application of sociology to the discussion around COVID-19 vaccines. Let me begin by making a relevancy claim. Some think this is just a medical issue or just a biological issue. And our previous speakers and colleagues have done an excellent job of illustrating that's what it appears to be. But viruses are spread socially from person to person. Vaccines are a strategy to mitigate, limit, or stop the social transmission of a virus. So this is a social issue as much as it is a medical or biological issue. And virtually every public health mandate up to the arrival of the vaccine has been social in nature. Let me give you 3 theoretical lenses and illustrate what I mean. The first is structural functionalism. If you haven't taken Sociology 101, don't think this counts as taking it. You still have to take it, but you're going to get a little bit of a thumbnail version of it here. Structural functionalism is a positivistic approach to the study of society that looks at the institutional influences of human behavior. Parts of a society are working well and they make the society function well. When the parts don't work well, it can be dysfunctional. Talcott Parsons, a mid-20th century American sociologist interested in the stable functioning of society, saw sickness as potentially leading to societal breakdown because the sick could not fulfill their necessary social functions in society, such as parent or worker. Therefore, sickness, according to Parsons, must be managed and given special status in society. Parsons termed the phrase sick role as having both individual rights and social obligations. The sick role had both a personal, individual, and a group social component. First of all, the two rights of a sick person. First, the sick person is exempt from normal social roles. If you're going to miss an exam, you better have a doctor's note. If you're not going to school, you better be able to prove to your mom or your dad that you're actually sick. You've got to legitimate that you're sick in order to be excused from your normal roles. The second right of a sick person is that they're not responsible for his or her condition. Influenza, a cold, a broken leg are considered the result of misfortune, not personal will or desire. Therefore, sympathy rather than blame is considered the appropriate social reaction of others. But the sick role also has obligations, social obligations, according to Parsons. The sick person should try to get well. The sick person is duty-bound by society to want to get well. The sick role is only temporary. Thus, if a person receives a diagnosis of pneumonia, he or she must do what the doctor orders, or the legitimacy of the sick role deteriorates into labels of shame, like carelessness or foolishness. And then finally, the sick person, this is the second obligation that sick person has, they should seek technically competent help and cooperate with the physician. From the viewpoint of Parsons, illness is a form of deviance. It is a potential threat to the social system unless it is managed for the benefit of the social system. Medicine is the institutional response for providing legitimacy and justification and for bringing the sick back to wellness. Medical institutions can be seen as agents of social control in much the same way the church once was. Second theoretical lens, conflict theory. It has similar interests as functionalists, except they challenge the fair distribution of those individual rights and those social responsibilities and highlight the economic inequalities that marginalized populations experience compared to elites. It's a critical approach to the status quo in society. In a critical theory, all social arrangements have political or economic bases and consequences. Conflict theory tends to focus on gender, class, power relations, and dynamics. This might be illustrated at the individual level by a queue jumper who pays in order to get vaccinated earlier. They want preferred access to the vaccine. Or it could be a privileged member of society who chooses to be a free rider as a reason for not getting vaccinated. At the macro level, we could look at how the nation of Israel arranged early and professional access to the Pfizer vaccine in exchange for comprehensive population level data. Israel now has over 60% of the entire population with at least one dose of vaccine. Yet at the same time, the Gaza Strip and West Bank are much further behind and have access only to doses through the International COVAX Vaccine Sharing Scheme. We could also look at the economics regarding the pharmaceutical industry and the pros and cons on an increasingly medicalized society. I won't get into all of the details here for the sake of time, but Moderna, in conjunction with NIH, actually proposed their mRNA 1273 2 days after the Chinese released the genome sequencing. If I were looking at a pandemic through a conflict lens, I would focus on what I would describe to be what I think is going to be a dark legacy of the pandemic. We will want to focus on the positive sides for sure, the resilience of the world's populations, that rapid development and deployment of the vaccine. But behind all those successes will be the unforgettable tragedy of how our nation's elderly ongoing victimization was exposed by COVID-19. I would point to the fact that almost 60% of long-term care homes in Ontario are run for profit, 35% in BC, and that these for-profit LTC homes were associated with even higher levels of resident deaths than the publicly run ones. Finally, let me turn to the third major theoretical school of sociology, symbolic interactionist. Symbolic interactionist sociologists study how the subjective definition of social realities are constructed, and how this reality is experienced and described by the social actions. Symbolic interactionists are interested in the symbolic nature of social life. What do you mean by this action or by those words? Is your behavior or your action purely functional, or is it trying to convey a larger message? I could talk about both the social and mass medias as messaging platforms, what they promote, what they cover, what they suppress, or what they ignore. We could look at the almost religious behavior of people who both support the widespread use of the vaccine and those who critique that approach. Instagram and TikTok videos about people celebrating the vaccine are often with supporters cheering them on in a type of baptismal parallel. The vaccine has been characterized as a savior. Likewise, individuals and groups critiquing the rollout gather for collective effervescence, often accompanied by homilies in support of their cause. They may use terms like, "This is the end of the world as we know it." And both groups claim true devotion to what sociologist Max Weber referred to as the modern high priest, the scientist. I could talk about the social shaming and the stigma that may be involved with being invited to the front of the vaccine line because of your obesity or other pre-existing conditions associated with heightened COVID risks, or the ad hominem labels like anti-vaxxer. One only has to look at the pro-choice label to see how derogatory the term anti-vaxxer comes across to those who may have legitimate concerns. I'm out of time, but let me leave you with a final example of a symbolic interaction illustration, and hopefully a way to return to the uplifting faith part of this talk. On January 30th, 2021, I had the opportunity with several of you to hear Francis Collins, director of the National Institute of Health and committed Christian. The event was sponsored by the Canadian Scientific and Christian Affiliation, in which Dr. Sikkema is the executive director. Dr. Collins spoke about the creation of God in relationship to the recent developments and deployment of the COVID-19 vaccines. Most of the technical details went right over my head, but there was a piece near the end of the talk that I cannot get out of my mind. In answering a question regarding people with vaccine hesitancy, he did not ridicule, he did not label, he did not throw anyone under the bus. Instead, he provided what I considered to be a mature, graceful, compassionate, Christ-filled answer. He said that those who are hesitant about the vaccine are often those who have not been well served by the medical system. They are often disadvantaged groups who have been mistreated or even victimized by traditional medicine, often in the name of science. He mentioned people of color in the Tuskegee syphilis studies. If you're not familiar with that, you should look it up. Indigenous groups and their poor treatment by the establishment. I could add how women have been marginalized or patronized by a male-dominated medical system. He went on to say that we need to understand the circumstances that lead to this hesitancy and meet people where they are at, rather than judge and condemn them for valid concerns and honest questions. With that answer, I saw an individual supporting the needs of society while also recognizing the needs and concerns of the marginalized. I saw the Trinity at work. I saw Christ at work. Thank you.
Thank you, Todd. I believe it's me again for a slide or two before we go into the question and answer, and I just need to find the button to share my screen. There we go. I only have a few brief points to make here, and I'll make them briefer in light of the time. The vaccination issue is a sociological issue, as Todd has nicely spoken to. And if it was just as simple as the science, yeah, but it's not. And as Christians, speaking as a Christian, we happen to be in communities that are often perhaps more hesitant on average, or have more concerns on average. So just a few thoughts, not that I claim any great expertise here, but just a few thoughts about a Christian approach to vaccination. The first point to make is that vaccines are safe. Now, any medical procedure of any kind will have a low level of risk. And the purpose of the trials, like I mentioned before, the phase 3 trials and the post-trial, post-approval monitoring, those are designed and intended to minimize that risk. So getting a COVID vaccine is safer than crossing the street at this point. And as a result of that, Christians should be encouraging our fellow believers and people in general to get the vaccine. For themselves, as Todd nicely spoke to, there's a benefit for ourselves in terms of reduced risk of illness or hospitalization, but there's also a benefit for our neighbors and for the vulnerable. If I don't get a COVID vaccine and I get COVID, and if I happen to be one of the few that needs to have an ICU as part of my treatment, then I would be taking up an available space that someone else might need and might need more desperately than I. There are some people who cannot, because of their medical conditions that they have, cannot have vaccines or will not develop an appropriate immune response as a result of getting a vaccine. So those individuals actually depend on enough people around them being immune through vaccination, such that the virus will not transmit through a chain of infections to them. This is what is called herd immunity. And immunocompromised people are protected by those of us that are capable of being vaccinated. So the vulnerable and the weak and those with immunocompromised conditions are depending on those of us who can be vaccinated to be vaccinated, because even if COVID isn't a big deal for me, perhaps it could be very, very serious for someone who isn't capable of getting the vaccine. There are also some ethical considerations with respect to COVID vaccines and vaccines in general, actually, that some Christians have highlighted recently, and that is primarily around the use of fetal cells and concerns about basically the cells derived from aborted fetuses. There's a very excellent webinar that Francis Collins did that touches on this issue, and I could refer you to that. But maybe I'll just briefly say, COVID vaccines do not contain the cells of aborted fetuses. COVID vaccines in some cases are tested on cell lines, so replicating cell lines, basically cells that have been replicating and made such that they will continue to replicate, that are ultimately derived in some cases from aborted fetuses from the 1970s. So some Christians do have that concern, and it's natural for us to say, you know, I don't like that. I don't like that that's the case, that these cells in any way might have been derived from an aborted fetus, and that they might be used in some way for medical research. Those cell lines are used for a large number of medical research applications, and not just vaccines. So whether we are aware of it or not, we actually depend on a lot of the technologies that are produced by those. Now, new fetuses are not being aborted, new cell lines are not being set up. There are regulations that prevent that from happening. The fetuses that are the source of those cell lines were not intentionally aborted for that purpose. Those of you that might be familiar with a well-known book called The Immortal Life of Henrietta Lacks. This is a woman of color who, unbeknownst to her, had a biopsy taken and a cell line developed from that biopsy. And that cell line is still with us today and is being used. It's in some ways a similar situation to that. These individuals, these babies that were used to produce these cell lines, and Henrietta Lacks as an example, were not able to give consent, and these cell lines were taken against their will or without their knowledge. And yes, there are ethical concerns about that. Absolutely. It is my opinion, and it's the opinion of Francis Collins and many other Christians in the biomedical sciences, that the use and the vaccines that we have and how they are developed is not an ethical dilemma for Christians. We could talk more about that during the Q&A if you want to. That is an issue of concern for many Christians. Todd mentioned Francis Collins. I, of course, will mention Francis as well, because he founded the BioLogos Foundation that I worked for many years, and I've had an opportunity to work with him over the years. And Francis is the director of the National Institutes of Health. He's a very prominent person as a result of that in world leadership with respect to vaccine development in COVID vaccines. Francis is a dedicated Christian. He has never been shy about his faith. And he's one of the very few people who have transitioned between different presidents as different administrations have come and gone. So he was appointed by Obama, Donald Trump kept him on, and Biden has kept him on as well. So he will now serve under his third president. And part of the reason for that is that he is so well respected as a kind, compassionate voice, and also as someone who is a scientist of the highest caliber. So Francis is also of the opinion that COVID vaccine should not be an ethical dilemma for Christians. But it's okay to talk about it. It's okay to find out more about it, and it's okay to ask those questions. We don't serve anyone well by avoiding those questions. So those are my brief thoughts that will perhaps prime the pump for our later discussion.